Let me see.
Well, it was the first time that I collaborate with scientists in a non-medical way. And I had a lot of question marks about it because I thought about how they were going to handle someone bursting into their environment. I know that it's very difficult. I reflect a lot on the idea of the scientist or the artist. What does the position of the artist and the scientist and the power they have, what does it mean and where is it located? The first project is I take a bioluminescent strain of bacteria and it's ordered to commit suicide. Okay, so it has an electrical component, a programming component, and a chemical component. The voice recognition software is a basic voice recognition software that gives a text command. The keyword is death. And then the text command will induce a pump to release a chemical that will basically cut the DNA of the cell. And this is how the bacteria will die. The second project is taking a bacterial strain from my feces, transfecting it with a bacterial DNA that contains a neuropeptide Y gene. So neuropeptide Y is a molecule that has been linked to protection against PTSD, post-traumatic stress disorder, and to talk about, hypothetically, flushing it in the wastewater system of Beirut through the toilet. The strain, speculatively, will regain the water cycle and go back into drinking water. People will no longer be, will be more resilient. They can adapt to change better, and they'll be less prone to post-traumatic stress disorder. And in the region, post-traumatic stress disorder is a big issue. So it combines a bit all these ideas together.
Okay, very good. So, well, you know, this is a typical case of an artist that comes to one's laboratory and then she gets inspiration. So I don't know if you understood everything she said, but basically she proposed two things. One of them is to have this system through which you say to a culture, commit suicide, and then bacteria activate some, I mean, you have hardware, or a pump you have here and there, and then a voice processing interface will open a valve that will add an inducer for a killing system, okay? And then cells that are otherwise luminescent will lose light emission and will die, okay? And the other is about engineering a bacterium out of her feces that is engineered to produce a hormone that is known to endow people with optimism. And then her idea was to go back to Beirut and then put this bacterium through their water treatment network, and then eventually bacteria will go into the entire population microbiome. I insist these are not real, okay? So I don't think that we have done that because they would be in big trouble. But this conceptually, you know, it indicates as a scientific or an artistic exploration of what could be done with synthetic biology. Okay, so then, you know, one thing takes us to the other. And then rather than perpetuate the present and to think, to live in the world as it is, can we think in a different future? So I'm going we're not going to make this break, you know, because I would like to get to the end of these stories I want to tell you. And so architecture. So architecture, what is architecture? You go out, we see these buildings, squares and so on, and then we take for granted that everything in architecture has to be as we know it, right? But maybe we can think differently. For instance, it has been known for some years that some bacteria are able to accumulate on their surface some calcium carbonate, okay? So this some calcium carbonate can heal concrete cracks, you know, and this is one of the most, say, attractive developments of synthetic biology to some extent, because this allow us, or will allow us in the future, to develop a concrete that will self-heal when it cracks, okay? And well, there are many bacteria that can do that, Bacillus and other bacteria, And then the vision is that if you make a product, a construction product, that is a combination of say, bacteria with this concrete material, with these concrete solids, then this is a system that will last forever or for a long time, because simply every time that there's a crack, the bacteria will fix CO2 and will put these, materials to fill the gaps, and at the end, the concrete can self-seal. Okay, so this is something that would be very nice. And there's an intensive research in this type of things, because the idea is that you have a crack in the concrete, then the crack can be colonized, the part of the crack that is to the outside, by these bacteria that do the job, and at the end the bacteria can produce some healing calcium carbonate, and then you fix the crack here. So this is something that in terms of construction technology might make a big difference. But there's something that I still find more inspiring, that is the following. Plants, as any other biological object, have a shape, and the shape responds to a genetic program. Okay, so if we look at the life systems, we have animals, we have insects, we have jellyfish, we have worms, you have all types of things. And at the end, the shape, the physical shape of everything is genetically determined. And as it is physically determined, chances are that by modification of the genetic program that originates this morphological diversity, we can generate forms and shapes that otherwise would not happen, right? So one of the things that are determined in particular are plant roots. Okay, so plant roots, this is Arabidopsis, so it's a model system, but it has been known for a long time that the shape of the roots is genetically determined. So depending on whether you have a gene, a mutation here, or a combination of mutations, or you put a plasmid or whatever, then the roots can be small and curly, it can be long and linear, they can wave a little bit, they can branch out at the beginning, they can branch out at the end, they can have kicks, and so on and so on. And the shape of the roots is genetically determined. So on that basis, in the last few years, there has been very interesting research on reprogramming the shape of the roots by using synthetic biology and by rewiring some of the genes that are involved in the morphology of the roots. And the idea after that is to create root forms that are not natural, that are not the ones that are given by nature, that are different. Okay, so in some cases, this would be the situation, the standard situation, but then you can put these genes under the control of other inputs, and then you create an or not gene here, a gate, or you can have different gates here and there. And as we have discussed these days, the advantage of having logic gates is that the outcome or the output of some of the gates can be connected as inputs to other gates, and at the end you create a large diversity of situations. And you can make circuits and connect and so on. So there are various laboratories that for many years have been trying to look at the shape of the roots with the intention of rewiring the different genes that are involved in their morphology to create new forms of roots with different, say, shapes and different possibilities. So for instance, in this case, well, depending on the genes that you have, the different sequences that you implement, then the shape of the roots might be quite different. Okay, so why looking at, or why knowing and investigating and making these sequences that are determined genetically by different genes for their morphology, why it can be so important for the future of architecture? Well, this is a tree. This is a picture I took this morning in my way here. Probably you know it's an amazing tree. Well, I have to tell you something. You see this every day, and you may not be aware of some things that are absolutely amazing that happens just next door. Okay, and these trees that are biams, I understand that this is the name, do something that to me is absolutely extraordinary. Well, to me and to everyone, right? And is that roots, they have an initial root, but then once the tree goes up, then some additional roots start coming from the trees. They touch soil, they really become real roots. And then at the end, what started as a root hanging from a branch ends up becoming a pillar. And at the end, the pillar sustains the whole structure, okay? So what is behind this morphology, I think is unknown, because I don't think that anyone has really looked into this. But obviously, this morphology is not casual. It has to be genetically programmed, okay? So if this is the case, why don't we can think in, making houses with trees, okay? Program a whole house, a whole building with trees. So the vision, and this is a project that I'm running with a young architect in my country, could be, again, this is a theoretical prediction, and nothing that has been done so far. Well, you could start with a place where you want the house or the building to start. Then you have to inoculate the seed that obviously genetically has to be heavily programmed to release at the end, or to produce at the end the different shapes that you want to have in a house. And then you let it grow, and then the building would grow and grow and grow, following a pattern that you can program genetically. And then eventually you will have a kind of skyscraper that could interact with other standard skyscrapers, but that would be entirely organic, okay? So this building could be a part of the landscape, could replace the landscape or just be added to the landscape. And the idea is that in one case, the standard architecture is very concrete and steel and these things, whereas the biological architecture will be based on exclusively biological components, life components. This is not science fiction, but it's science anticipation. This will happen if we have the tools and we have the knowledge to do these things. Now, the idea is that these buildings will have, instead of having the square windows and the straight lines that are so characteristic of modern architecture, it will have a curve, say a standard, and it will work well as rooms or tables. We can program inside the rooms to have some forms and everything. This is something that is not absurd. I mean, it may be difficult, it's not absurd, because every developmental program is determined genetically, and therefore it is not impossible to think on creating new forms that we can implement in biological systems for our purposes. So, well, you can go one by one through all the different elements that you may have in these big buildings. You may have a room, and the room is illuminated through sunlight. And the sunlight might be collected from the outside, but it can be transmitted to, for instance, luminescent bacteria. And then you may have illumination by luminescent bacteria there. You may have also ways of exploiting existing ways of transportation that you see in plants. Maybe you can blow it up and then make a kind of elevator that is based on biology. The idea perhaps will be much slower than you have the elevators here, but at least conceptually would be possible that you go from one location to the other, because in plants you have material transfer from the roots all the way to very, very high on the basis of pure biological events. You may have parts of the building that will be illuminated by sun, and therefore you can have an internal garden inside the building. You may have also some way of escalating from the surfaces on the basis of some self-motion genetic device or some other biological component. So you can encapsulate yourself in a membrane and see whether the membrane can be transported. So some of these events are like scaling up something that happens at very low level in cells and so on, that cells can migrate from one place to the other, can open the membranes and communicate with each other and so. So why not thinking that we can get not only inspiration, but also recruit the corresponding activities to think on a new type of architecture that is entirely organic. Also, the fact that you have something completely live enables you to engineer the surfaces, okay? So maybe one of these surfaces can be coated with a type of bacteria that resists irradiation, for instance, as we discussed yesterday over coffee. So once you have the possibility to program DNA, you can do anything, absolutely anything. Obviously, in some cases, it might be easier than others, and we are still in a very, very incipient stage in the history of synthetic biology, but why not? I mean, this is something that is not impossible. Similarly, you can collect energy. I mean, you can do many, many possibilities. Also, from the point of view of architecture, we take for granted that the illumination of the streets have to be either with gas lamps or with, in the past, or with electricity, right? But why not? I mean, given that some bacteria produce luminescence, we can think on illuminating the streets with luminescent biological items, right? So you know that there are different types of luminescent bacteria that colonize. I mean, the ones that are best known are the ones that colonize some crevices and some, say, locations of the body of squids. And then they can go deep into the sea and they get this illumination. And some of these bacteria, in particular one Vibrio, let's call Vibrio harvey, that is the one that produces this luminescence here, can be taken to the laboratory. And if you go to a room that is dark, you immediately see light emission. I don't know you have seen this yourself, but I tell you, this is an absolutely amazing thing, that you go to a dark room, you take a plate and you see the colonies glowing. I mean, it's like a miracle, you know, it's really difficult to believe that this is happening. Okay, so then again, this phenotype, this property is determined by genes that can be programmed, can be subject to circuits, can be amplified, can be modified in various ways. So why not? You can think on illuminating cities with glowing trees, for instance. You can put the genes for light emission into a plant and program that the production of the plant of these genes or the activity of these things happen in the absence of sunlight and concentrate on the leaves, for instance. Okay, so people interested in molecular biology of plants or genetic of plants know exactly the genes, the cell types that are here and there, and therefore the possibility exists of making these trees. I mean, they have not been made yet, or they may have not been done yet, but why not? It's not impossible. Or you can think also on illuminating not just the streets with plants, but just having, instead of having electric lamps, to have bacterial lamps that produce electricity on the basis that produce illumination and light on the basis of purely biological procedures, right? Okay, so again, as we discussed with the cheese coming from the feet and these things, this looks weird. This looks like, you know, but why not? I mean, this is something that we should not stop our imagination and our creativity to think that this is impossible, because it may not be impossible at all. Another straight turn that I find quite interesting in this direction is the construction through synthetic biology methods of life materials. Okay, so life materials is, as its name indicates, is you know, this concept that you have either textile materials, other types of materials that are functionalized to do something else apart of just being a physical body. Okay, so depending on whether they have some starting physical characteristics, that they are flexible, they are breathable, and so on, then it is possible to engineer them either through combination with active bacteria or by engineering the product itself with some features that are recruited from bacteria, but then are not delivered by bacteria itself. And then you can have an entire set of possibilities here that, unfortunately, the resolution is not very good in this slide, but that can help you to really add some value to otherwise inert materials, right? So you, this is something that, for instance, in the textile world, and I will mention that in some detail, has been very, very successful, because you can have different types of textiles that, are part of just being used for covering your body, can give you much or many more interesting activities. They can wear biosensors, they can wear monitors, they can you know, make you feel warmer. I mean, they can be engineered to take advantage of the motion to generate energy. So there are many, many possibilities of engineering materials based on biological, say, starting points. So one of the most interesting starting materials for producing, say, this type of stuff is bacterial cellulose. So cellulose, as you know, is something that typically is produced by plants, together with lignin, lignocellulosic materials, and so, but there are many bacteria that naturally produce cellulose, either as nearly pure cellulose fibrils or in combination with other molecules. But the thing is that many bacteria, as I said, produce this polymer, that basically is a polymer of glucose in various conformations and so, but at the end what you have is a polymer of glucose that generates this material that we are all use, and is cellulose. Okay, so this is something that has been known for a long time, and there are possibilities also and a lot of say studies and data on how to recover this cellulose from the bacterial production say setup and then convert that in a layer. In reality, cellulose is produced by many bacteria, but not all of them produce cellulose to the same level and to the same quality. Okay, so the typical way of producing cellulose layers is by having a biofilm and then letting the biofilm to develop under conditions where cellulose production is stimulated, and then the biofilm is taken from the place it is formed, it is processed, and at the end you have a kind of film, more or less thick, that resembles many of the characteristics of textiles, in particular of leather. So this is how cellulose is produced in plants, and well, this would be a standard way of producing cellulose. But as I just said, some bacteria can produce this kind of slimy and jelly-like material that is cellulose, and once it is treated, then you generate something that is a starting point for derivatization and conversion into something far more useful than just plain cellulose. So this is a cup with kombucha tea. I don't know, you have kombucha tea here? Okay. Okay, kombucha is a type of tea. Maybe the name sounds like more Chinese, I don't know. That when you let it outside, then it attracts as a very interesting microbial community that forms a thick cellulose-rich biofilm. So this is something that has been exploited because the composition of this community is known, and therefore the possibilities of manipulation are considerable. So this type of cellulose has been used already to make some coats and some things. The texture, when you touch it, is similar to leather. I mean, very, very similar. And the advantage is that you don't use animals, and it can be produced at large scale with a very, very simple, say, bioengineering setup, okay? Because this is not produced by engineered bacteria or anything. This is produced just by naturally occurring biofilms that have been exploited by various companies, including one that, as I told you, one of the first days I participate in. And well, there are some fashionable, say, items that can be purchased that is based on this type of things. There are even shoes, for instance, made with bacterial cellulose. There's a paper that just came just a few days ago, in which there's this lady, Lisa Melton, that proposes that, well, some shoes can be produced on the basis of bacterial cellulose. And at the end, well, I don't know if you like this particular shape, that you know, but in reality, what the paper or this report demonstrates is that you can really do it, right? That you can generate something that looks like functionally will be like a shoe on the basis of materials that are exclusively produced by bacteria, not by animals. But given that in this bacterial community, in these biofilms, you have a bacterial community, the bacterial community has DNA, and the DNA can be programmed for specific purposes, okay? So in some cases, the main players of the biofilms are known. You have a yeast here, you have another bacterium there, you can put them together or not, and then you start generating data on the composition and the properties of the different types of leather that you may have produced as this supernatant, say, film that you get when you culture it in a liquid medium. And then the advantage is that you can, again, play with the genes that are involved in the creation of the biofilm, and you can add various, say, features here. And then you may examine whether some of the components of the biofilm are amenable to genetic manipulation, for instance, by connecting some of the genes and some of the promoters to some reporters, and so you can make, and this is something that has been made mostly by someone in the UK called Tom Ellis, and then you can monitor, you can see whether you can, say, adopt expression of some of the genes to formation, for instance, of a color. I mean, you can really start playing with the genetics, and when you have a system that will respond to one given molecule, we can replace the GFP by a metabolic pathway, for instance, and then have a leather that is enriched in the production of something that you are really interested. One of the easiest things that you can engineer in the bacteria that produce the biofilms is a color, because colors are easy to manipulate. So for instance, in this case, the idea was to have eventually a type of leather based on microbial cellulose, on bacterial cellulose, that is dark. And to get this color, you need to engineer a pathway on plasmids or in the chromosome with promoters, the terminators. I mean, similar to what we discussed yesterday about artemisinin, where you have the different steps in the production of this dark compound, this leather-like color, and then have that implemented in your favorite, say, biofilm here. And then in this particular case, what they did, and it's also very good, is to have the genes for production of this pigment responding to illumination. So the genes were engineered under the control of an optogenetic system in such a way that now you have the biofilm, you illuminate bits and parts of the biofilm with light, and then obviously the bacteria that are exposed to this light will respond to the inputs, and because it carries an optogenetic system, then only in those locations expression of the genes for color will be produced, right? Obviously, this goes together with making a circuit, with some feedback controls, with this and that, but at the end, the idea is that you may have a printable, a biologically printable material that will develop a pattern of your choice depending on the parts that you illuminated with, okay? So this is something that is quite cool. And this is what these people made. So depending on the conditions of the growth, depending on the illumination, and depending on the treatment, then at the end, they could get different types of leather-like things that in this particular case was developed later as a kind of wallet that you can to put your money or whatever. And then you can control on the basis of the illumination, the intensity of the color and so on, and you can develop patterns, and you can finally cut the leather with given, say, specifications and produce, in this case, another shoe, right? So this shoe probably at a distance will be difficult to distinguish between this and other types of say, shoes, right? Okay, so this really tells you that, again, we take for granted that leather has to come from animals. No, it doesn't have necessarily to come from animals. It can be produced through bacterial methods. And not only that, we can do with leather, like materials, things that original leather cannot do. Like, for instance, developing a color or developing patterns in the absence of chemical dyes, just on the basis of expression of pathways that will give colors here and there. I mean, the last screw turn of this endeavor to create textiles and to create materials that can be useful for different purposes than the ones that nature has done is this concept of what is called wearables. So wearables are, say, some type of films produced in many cases by bacterial, say, matrices, where you can embed given biosensors, okay? And the biosensors may respond to different types of, I mean, they can be based in different principles, and also they can be responding to different, say, inputs from your body. So for instance, the idea in this case will be to have a sticky little patch that you put in your body, and then the pad has been engineered with some devices that will respond to given signals. And the devices can be a constitutive promoter, no big deal. You can have a transcriptional regulation, you can have a switch, or you can have a ribo switch, okay? So you can play with every trick that we have been discussing these days for engineering gene expression and embed these type of tricks in your film here in such a way that when the film touches your skin, the skin will release some signals. These signals will be recognized by the biosensors. And then at the end, you have a kind of diagnosis of what is going on in your body, right? So there's this growing tendency to have tools for diagnostics that are based on these very, very simple kind of constructions where you combine genetic engineering, you combine bacteria, you combine devices that you get from different locations. And at the end, you have a product that has the power to give you a diagnosis about a given health condition. Okay, so this is something that, in my opinion, is really extraordinary. But we have to go back to some of the earlier, say, discussions that we had, and is what we think biotechnology in combination with synthetic biology will bring to us in the future, okay? So there are basically like two possibilities, and this is something that I leave to your consideration and also to your discussion later or after I'm done with my story here. So there is this idea. Well, I don't know. I will ask you later what is your reaction to everything you have heard these days and the possibilities of synthetic biology and all that. But there's this idea that SynBio is just a way of doing the same things that we used to do before in a different way. Okay, so if we don't want to have petroleum, no problem, we'll produce biofuels. If we don't have to want leather killing animals, no problem, we'll do it differently. If we don't want to have chemical industry producing high specialty chemicals, no problem, we have bacteria doing them. But if you look at it if you think about it, this approach involves that you want to maintain exactly the system as it is. It's just that you change a bit the way the manufacturing of products is made. So there's this lady here that is the CEO of a big company in the US, a big SynBio company in the US, that says, I mean, this is a tweet that appeared yesterday. So it's something that I wanted to bring because, as I told you at the beginning, synthetic biology is something that is happening right now. So it's not a thing of the past. It's not an entirely consolidated discipline. It's something that we are in the middle of it, the development of it. Okay, so if you read this, look at our traditional industry. SynBio is coming for you. Solutions are greener, more sustainable, circular, and most important, consumers don't have to change their behavior to utilize them. Okay, so this would be the notion that synthetic biology will not be I will not say revolutionary, but will be a different way of doing the same, a different way of making money, and a different way of interacting with chemical production of things, manufacturing and so, but basically nothing will change. It will have the same, we can maintain our society and it is, and we eliminate some of the downsides of the contemporary chemical industry, contaminations and so, but basically the rest will be the same. Okay, so I responded to this Twitter on this one, okay, and it says, well, is SynBio about changing everything for everything to remain the same? Or is it a deep breach in the habitual exploitative way we interpret with nature since the first industrial revolution? Okay, well, I leave it as a question, but obviously I don't think that SynBio should be just one more way of business as usual. In my opinion, the power of synthetic bio is really make a big difference in terms of our interaction with nature. Because if you think about it, we have this, at least in the Western countries, because of the different traditions, the way we interact with nature is exclusively through exploitation. The nature is there just for us to take advantage of it, exploit it, and make money as much as we can, right? Obviously, this situation has taken us to the crisis we have at the moment, and synthetic biology opens the possibility to develop a new way of interaction with nature that is not identical to what we had before. In the case of synthetic biology, more in terms of conversation, dialogue, win-win interactions, not so much in terms of domination. That is what has been the currency of our interaction with the natural world until now. So, well, I leave you with this question here, and well, you may have your own opinions that we can discuss. I will be very interested to hear them. But in any case, my kind of final message to the audience in terms of the lectures is that synthetic biology is not only a technology, it's not only a science, it's also not only a way of looking at biological systems, it's also a tremendous tool that, depending on how we use it, can make a big change in the way we interact with the natural world, and therefore it will be a big actor in the future of our society or our human endeavor, as you call it. Okay, obviously this story is not at the end. This is to be continued. I don't know whether it will be continued next year or it will be continued for years to come, probably, because I insist that the field is not settled yet. It's not like physics. It's not like chemistry. I mean, the field is very, very active. There are many opinions, many perspectives. Some of them are more biotechnological, others are more fundamental, others are, you name it. So if I give this course in 1 year or 2 years, probably the context will be different because it is a permanently evolving field. But in any case, I think it's super fascinating. And in fact, I started my career as a genetic engineer, and then probably I will finish it as a synthetic biology, because I think that right now there are very few things so exciting, so creative, and so appealing for talent as synthetic biology. Okay, so this is the end of the course. So I just want you to keep calm. Thank you very much for your attention, not only today but the previous days, for your patience with my not super optimal English. I hope that despite the fact that you may not have understood 100% of my words, at least you capture more or less most of the messages that I wanted to share with you. And also, for those of you that were not here the other day, I want to share with you again these coordinates that you are welcome to keep. And I re-invite you to keep in touch with me in case you are interested with all my coordinates here. I'm in the System Biology program, Centro Nacional de Biotecnología in Madrid, and my email, my phone numbers. Well, phone numbers are normally not used much these days, but then you have this webpage here, and then you have this Twitter account that you are welcome to follow or not, I don't know. And you have this lab video. I cannot resist the temptation to show you the lab video, and just as a final kind of show, and you will see the type of things that we are doing in my laboratory. Okay, so this is right. Well, it doesn't have a sound, so this is something that you will see. But I would like to share a video that we have on what we do. Let me see. Well, there are some videos there. Where's the video here? Okay, let me find it. Sorry. Okay, so what I will do instead is that I will send you the link to this video. We have a kind of explanatory video of our interest, what we do, the people in the laboratory, and so you have a real, say, view of how a synthetic biology laboratory looks like these days. The small video I show you from Lara is also an indication. I mean, everything was taped in my laboratory, recorded in my laboratory. But basically all laboratories look the same. So it's not that you are going to find especially exotic instruments or whatever. But in any case, you have a picture where I work and so on. Okay, so I think that was basically all I wanted to tell you, not only for today, but for the rest of the course. So now, in a while, we'll go for lunch, those who want to join, and then we'll return by 2 or so, and then we just have this final discussion. Remember that you have to give your presentation. I will have this presentation of IM or IM-like projects, okay, and see how it looks like. And also, I would like to ask you after lunch, to please give me some feedback, So Ashmita told me that you are supposed to fill a form or something on the feedback to the course. Okay, well, in any case, but even if you have a form, I would like to get your feedback directly. And the answers, the questions that I want to ask you, and I would please ask you one by one, what your reaction is, whether you have learned a lot or nothing or a little in this course, and what type of, what are the things that you consider like more interesting of the whole course, and also what are the things that you consider the least interesting. Okay, so some classes were like, I can notice that because, I'm talking here, but I have your vibrations, so I noticed that some classes were very interesting for you, others were a pain in the neck. So I think that I would like to identify which ones were more interesting, what others were less interesting, because as I told you, I'm planning to write a textbook, and your feedback in that respect would be very, very interesting. Okay, great. So that's all for today and for the formal part of the course. Thank you very much for your patience and all the rest of it, not only to those of you, but also to those that have showed up occasionally to the course. And I just wish you the best. Now, let's see. Do we know where we're going for lunch? You will guide us, or Ashmita will tell us. Okay, very good. Okay, so let's make a little break and let's wait for Ashmita to show up, and then we'll go for lunch, okay? Very good. In the meantime, if you are going to make a presentation or something, please make sure that your presentation is in the computer. Did you do it already? Okay, please start loading your presentations. So at the end, we catch up with the time, okay? Because the session in the afternoon will be shorter, I hope, than the session in the morning, okay? So the more we advance in the morning, the less time will we around in the afternoon. Okay, here we go.